Here's what I mean by beyond CRISPR-Cas9. Typically, when you think about CRISPR-Cas system, you think of Cas9-directed double-stranded DNA cleavage. But in reality, Cas9 is not a solution to all problems. For that reason, we have variants of Cas9. We also have alternative Cas enzymes like Cas12, which also targets DNA but works slightly differently. There's Cas13 if you want to do something like RNA interference. There's also Cas12f, which likes to munch on single-stranded DNA. We'll talk all about this and many more examples, including the concept of collateral cleavage and alternative spacer flanking sites. Let's start with something familiar, the Cas9 family, which uses guide RNA to cleave double-stranded DNA. The workhorse, Cas9, comes from a bacteria called Streptococcus pyogenes. I've discussed the mechanism of Cas9 function in a separate video if you want details. A quick reminder is that it has two distinct nuclease domains that cut the DNA. It relies on a single guide RNA, which is scaffolded by 80 to 100 nucleotides. The nuclease cuts 3 nucleotides upstream of the PAM sequence. The PAM is generally NGG, which theoretically you hope to find every 8th position, but empirically this ends up being every 42 positions. But if you think about genomic regions like CPG islands, you will find a lot of these NGG sequence. But if you go for AT-rich regions, it may be difficult to find an NGG sequence. So SpCas9 is awesome, but at some level, it does show a fair bit of off-targeting. The cut it makes in the target is blunt, and the enzyme itself is quite a bit chunky. There are some engineered variants which improve the natural activity of the enzymes. Alternatively, you can find Cas9 in other bacterial species as well. Cas9 is one such enzyme. It exhibits lower off-target activity. Interestingly, it cuts in a non-blunt fashion. An engineered variant called Spot-on is also based on this. Like this, you have SpRY-Cas9 from another bacteria which has similar PAM requirement, but it is significantly smaller in size. SA-Cas9 is another one, but its PAM requirement is a bit too strict, but also pretty small in size. There are high-fidelity variants of this SA-Cas9 as well. These small Cas9s are nice because if you're trying to make protein fusions, for instance, a base editor, the small Cas9 size is a nice benefit to have. Based on SpCas9, there are some very impressive variants as well. For example, the HyperAccurate Cas9, which is mismatch intolerant and gets rid of most of the SpCas9 off-targeting activity. X-Cas9 is another engineered variant of SpCas9 that can recognize different PAM sequences, but it is still somewhat restricted to some preferential sites. To overcome all this, other Cas9s like SC-Cas9, which only really needs a G as a third PAM nucleotide, can also be used. Alternatively, there is SpRY-Cas9. SpRY-Cas9 does not have any PAM requirement, although it shows some preference for some sequences. SpRY-Cas9 is also used for restriction of large fragments when cloning gets difficult. You'll sometimes see it in hi-fi cloning methods. The Cas12 family is another DNA endonuclease. Interestingly, the spacer or guide orientation in Cas12 family is flipped relative to Cas9. The guide RNA scaffold is also less than half in size. The reduced size is mainly because the scaffold does not contain any tracer RNA. It is processed as one single fragment. The Cas12 PAM sequence is biased for T-rich nucleotides. And interestingly, Cas12 enzymes have one RuvC domain that cuts both the DNA strands on the target DNA. The cut is generally about 20 nucleotides away from the PAM, very different features compared to the Cas9. Cas12a, or CPF1, was the first enzyme in this family to be discovered. It recognizes a T-rich PAM and cuts DNA with higher specificity than Cas9, and it generally gives you sticky ends. And you will find that generally people use some engineered Cas12a variants instead. Cas12b is another enzyme in this family, which has a relatively relaxed PAM requirement. Everything else is similar, but the enzyme is quite a bit compact in size. 12e is another enzyme in this family with similar features and small size, although its variant called CasX is a little more popular. Just like CasX, there is CasY, which has a different PAM requirement and comes in a smaller size. And just like these, you will find there are many more Cas12 enzymes which can cut DNA. These are typically favored over Cas9 if you're targeting 3' UTRs or any AT-rich regions. Now, there's a separate family called Cas13 enzymes, and they target single-stranded RNA instead of DNA. This means you can now target the transcriptome, including mRNA, tRNA, or ribosomal RNA. The scaffold RNA for Cas13 enzymes is even more compact. And because this is an RNA endonuclease, its catalytic domains look nothing like RuvC or HNH. Interestingly, the PAM sequence for Cas13 is a single nucleotide called Protospacer flanking sequence, and it is located on the target RNA and can be any of the 4 bases. This essentially means that the edge of the target RNA should not be a hairpin or the literal last nucleotide, meaning that the edge of the target RNA must be accessible. Cas13a is the most common enzyme in this family with no specific nucleotide preference, and it has a very high RNA specificity. In single-strand cleavage, the concept of sticky or blunt cut doesn't really mean anything. It's just a cut. But these enzymes do have something called as collateral activity, which I will explain in a moment. The rest of the enzymes in this family are similar, with higher or equivalent specificity with variable levels of collateral activity and different enzyme sizes. So, what is collateral activity? From the RNA perspective, when a Cas13 enzyme binds its target RNA and makes a cut, it is possible that there are other RNA molecules floating in the space around it when the cut is made. So, as soon as the cut is made, the Cas13 goes into a frenzy and starts cleaving all the RNA around it. So, collateral cleavage is a random short-range or short proximity-based RNA cleavage where non-specific and random RNA is cleaved when a Cas13 engages its specific target. On a similar tangent, I want to talk about Cas12f. It's not a family enzyme, just one lonesome Cas12f. This one loves cutting single-stranded DNA, works on the same general principle of a compact scaffold, but under some specific conditions, it can also cut double-stranded DNA. Since it is cutting DNA, it relies on one RuvC domain. Unlike single-stranded RNA-cutting enzymes, Cas12f does not care about any sort of protospacer adjacent or flanking sequence. But just like the RNA collateral, Cas13f also deals short-range DNA collateral damage. The last one on our list is a special Cas enzyme called Cas3, which comes as a part of a larger complex of proteins called Cascade. This complex contains helicases, nucleases, ATPases. Typically, Cas enzymes are just nucleases, but this special one has helicase and ATPase subunits, meaning that this Cas can move, and like Pac-Man, this enzyme will shred DNA. It is a double-strand DNA-specific enzyme complex, and once the cut is made by the nuclease, the helicase unit gets activated, and it starts this unwinding process, and the nuclease starts eating all the DNA as it moves, essentially behaving like an exonuclease in a 3' to 5' direction. I made this video to show that the field of CRISPR systems and genome engineering is evolving very fast. There are so many things to talk about. For instance, a group of researchers in China engineered a Cas12a, which we discussed is an RNA-guided DNA cleavage enzyme. But this group in China reversed the specificity and published a variant that is guided by DNA and instead cuts RNA. Here is another cool discovery. When you think about CRISPR systems, you think about enzymes guided by a single guide, and there's a single cut location. Well, Feng Zhang's group at MIT discovered an orthogonal system called TIGR-Tas, which relies on 2 guides. It is completely PAM-independent system, very compact, and theoretically more precise than the Cas system because it depends on 2 guide RNAs. And as I said, this field is moving rapidly, and I hope this video gives you some idea where things stand at the moment.